Hi guys, let's do section 4. All right, we're going to be talking about how DNA becomes a gene. Okay, so the process of getting genetic information from the DNA into a functional protein. The first step is something called transcription. This is when DNA serves as a template for the production of an RNA molecule. And so basically, the DNA will unzip and it will provide a template for messenger RNA to be formed. Loose nucleotides in the nucleus will bind to the exposed DNA using Chargaff's rule. C's will bind with G's and A's with U's. When the entire gene is transcribed into mRNA, the result is a pre-mRNA transcript of the gene. So it's like the basic version of it. The base sequence in the pre-mRNA is complementary to the base sequence of the DNA. So it's basically a mirror image of the DNA. So in the first step, a single chromosome consists of one very long molecule with hundreds of thousands of genes. Okay, but we want to only transcribe or make a transcript of one of those. Okay, so we only want to make one protein, so we only need to transcribe and translate one gene. So the genetic information in one gene describes the amino acid sequence of one protein. The information in the base sequence of one side of the DNA molecule. That's the sense strand. Only one side is ever transcribed. Okay, the other side of the DNA molecule is just ignored. And the gene is the functional equivalent of a sentence. The segment of DNA corresponding to a gene is unzipped to expose the bases of the sense strand, of the strand that will be copied. The genetic information in the gene is transcribed or rewritten into a messenger RNA molecule, into an mRNA molecule. I like to think of it as like using Google Translate to take an article that is written in French and transcribe it into English so that it can be easily read. The exposed bases in the DNA determine the sequence in which the RNA bases will be connected together. An enzyme called RNA polymerase connects the loose RNA nucleotides that are floating around the nucleus together to make that RNA molecule. The completed transcript contains the information from the gene, but it's a mirror image of it, or it's a complement to it. So here we have a diagram of the process. You can see the DNA is unwinding, and RNA polymerase, which is this kind of clear-ish enzyme right here, it is bringing in RNA nucleotides and bonding them in the proper order to make a copy of only one side of the DNA molecule. Now this RNA molecule is not complete, it must be processed. So the pre-mRNA is modified or processed before it leaves the nucleus. The modifications include a cap on the 5' end. Okay, it's basically a modified guanine, which helps the ribosome figure out where the beginning is. Where is the start of that molecule? And then a poly A tail of about 150 to 200 adenine nucleotides is added to the end. Okay, this helps facilitate the transport of the mRNA molecule out of the nucleus, and it keeps the molecule from breaking down inside the cell. Pre-mRNA consists of two types of information, exons and introns. Exons will be expressed and introns occur between the exons. The exons are expressed and the introns are just in the way. Okay, this allows the cell to pick and choose which exons go into a particular sequence of mRNA. So a process called RNA splicing is how we get rid of those introns that are in the way. So an enzyme called a spliceosome will actually go in and cut out the introns and remove them so that only the parts of the mRNA that need to be expressed will be expressed. In prokaryotes, introns are removed by self-splicing. basically the intron has the capability of enzymatically splicing itself out of the pre-mRNA. It's able to recognize, hey, I don't need to be here, and it will actually take itself out of the molecule. So here we have a picture of a strand of DNA. The dark blue parts are the exons. The light blue parts are the introns. They are copied. They are transcribed, rewritten into pre-mRNA. The 5' cap and the poly A tail are added. And then the spliceosomes come in and they cut out the introns, the unneeded information that is just in the way. The introns are removed, and now we have a functional messenger RNA strand, which can then leave the nucleus, go out into the cytoplasm, and be the directions for making a protein. Functions of introns. So as organisms develop and become more complex as they evolve, the number of protein coding genes does not keep pace. So these are basically chunks of DNA that used to do a job, but they don't anymore because a sequence of DNA that does it better came along. The proportion of the genome that is introns increases as organisms become more complex. So humans have a lot more introns than a crayfish. Okay, possible functions of introns. Exons might combine in various combinations, which would allow different mRNAs to result from one segment of DNA. Introns might help regulate gene expression, and they may help encourage crossing over during meiosis. So scientists are still studying this, and they're still figuring it out. So back to the jellyfish. The green fluorescent protein, or GFP, is a bioluminescent protein found in jellyfish. Basically, it's what makes them glow. The jellyfish is normally transparent, but when it is disturbed, it releases aquarin, I think I said that right, which glows a green color. Scientists were able to isolate the fluorescent protein and develop it as a molecular tag. So they can basically put it into another organism so that they can easily track it. The GFP gene is inserted after the promoter region of another gene, which is basically the spot that says, hey, start making a copy here. Okay, so it's stuck after the promoter. The gene is inserted there, and expression of that GFP gene allows the organism to glow. These molecular tags can reveal cellular location and distribution of protein within a cell. So by inserting this GFP, we can see where that gene actually goes after crossing over occurs. So here we have some fish that have had the bioluminescent gene inserted into them. Okay, and you can see that it is causing them to glow, to fluoresce. And that's what I got for you guys. If you have any questions, let me know.